Hello everybody. So today I am going to deal with a fresh topic, and that is the identification of recombinant phages. We all know phages are the viruses which infects bacteria. Say for example Lambda phage, T even phage or T odd phages series are there. But here we are talking about recombinant phages. What are actually these recombinant phages? Suppose I show you the diagram of a common bacteriophage which has a head and a tail. So this is the DNA which is found inside this phage. This is the phage DNA. So if we remove out this phage DNA from the phage by certain techniques, and then we insert our gene of interest in this DNA. So this is the phage DNA. Then the gene of interest is inserted in it. So this will be the gene of interest. Gene of interest. This is the phage DNA. Now both of them are joined together at some appropriate sites. Say for example this is alright. So finally because when we have removed out the phage, we take a phage again here, and through appropriate tactics, I am not telling you the tablets, this DNA, which is a connection of phage gene of interest in the phage DNA, is inserted back into the bacteriophage, and then such phages which have the gene of interest in them are called as recombinant phages. They are called as recombinant phages. Alright, so this is the just an outline I have shown you, but exactly details how we remove out and insert the gene of interest in that I am not entering into that detail right now. And finally this phage is infected, and we have a bacteria here. We know the phages infect the bacteria. This phage infects the bacteria and then release its DNA inside the bacteria. So this is the infection process. Okay, let me draw a proper phage. So this is the bacteria, and this is the phage which is infecting this bacteria, and then the phage DNA is then released into this bacteria, and this bacteria will now multiply. The bacteria will multiply and will form a colony on an agar plate, on another agar plate. So this is the agar plate. Agar plate, and it is what we call it bacterial culture. Alright, so when this phage has infected the bacteria, it will cause certain changes in the bacteria, and the bacteria may undergo lysis, okay, or some other changes occurs in the bacteria. So if these are the bacterial colony, some of the bacteria which receives the gene of interest will be transformed, and then these phages will be formed inside this bacteria. Phages will be multiplied. The phages are seen in the bacterial colony. So this is the bacterial colony. This is the bacterial colony. So these are the plaques of the phages. These are the plaques which represent the phages, and we have to identify among these plaques which are the recombinant phages and which are the non-recombinant phages. The plaques are formed by the phages, because we know when this bacteria will be cultured, this bacteria is cultured, the phages will be multiplied inside this bacteria, and these phages will form separate colonies in the culture plate, and these colonies of the phages are called as plaques. And in these plaques we are supposed to identify the transformant plaques or transformant phages, which are recombinants and which are not recombinants. And we know the recombinant phages are those which have the gene of interest in them. All right. So we have got many methods to identify the recombinant phage. So these methods I am going to discuss one by one. So let us give our heading: methods to identify the recombinant phages. Methods to identify the recombinant phages. The most common methods I am discussing here. The first method which is utilized to identify the recombinant phages is insertional inactivation of lacZ gene. It is insertional inactivation of lacZ gene. This is a special gene. We know about this gene. What we notice here, almost all the phages and also Lambda phages and also M13 have got a gene in their genome, and this gene is lacZ gene. So this lacZ gene is found in the genome of the phages. Let me label this phage as Lambda phage. Okay, so what do? We take out this DNA. With this DNA, we take out this DNA. We cut the DNA through some restriction site, and this lacZ gene is disturbed. And then we insert the gene of interest in between the lacZ gene. All right. So we say that this is the DNA. This is fresh DNA which has been formed, and here we have inactivated the lacZ gene. So we can write here the lacZ gene is inactivated. All right, and we know the functions of this lacZ gene. This lacZ gene forms an enzyme beta galactosidase, and this beta galactosidase turns certain substances like xgal into blue colored substance. So this beta galactosidase converts xgal, that is a chemical substance. You can find about the xgal in my other videos. So it forms, this beta galactosidase turns the xgal in the blue color. Okay. So but if this gene is disturbed here, it has been inactivated, then the blue color will not develop. All right. So this DNA is inserted into the bacteria. This is the phage. This is the phage here. Now this is inserted into the phage, and we say that this phage is a recombinant phage. Alright. Now this phage will go and it will infect the bacterial cells. This phage is going to infect the bacteria, and then the phage will replicate inside the bacteria. The phage is then going to replicate inside the bacteria. During the replication, some of the phages will receive the recombinant DNA while others will not receive the recombinant DNA. Alright. So as I told you previous on my just now, that these bacteria will multiply and will form a colony. So for example, this is the agar plate culture. Agar, it is agar medium, we have taken the agar. This is bacterial culture. Now in this bacterial culture, the phages are also seen, because the phages, when the bacteria divides, the phages multiply inside the bacteria and then they rupture the bacteria and come out. So the plaques of the plaques of the phages will be seen, and we notice that some of the plaques are there which are having blue colored phages, while others, I can show it with the yellow color, they are colorless. That means they are not developing into blue color. And this black color is the lawn of the bacteria. That means bacteria are there, bacteria are continuously multiplying along with the phages. Alright. So we say that these plaques, the blue colored plaques are there, the white color plaques are there. So by analyzing the color difference, we can find out the recombinant phages and the non-recombinant phages. So this is blue. We can say that this is a normal phage. It has the colony of the normal phages. The lambda phages which are developing in these colonies or in these plaques are normal, the blue plaque. But there are certain other plaques which are white in color. I have shown it with yellow. White plaque. These plaques have got recombinant phage. So by analyzing the color difference of the plaques, we can identify the recombinant phage. The blue colored plaques are normal because they have the intact beta galactosidase gene. The beta galactosidase gene in them, these blue colored plaques are not inactivated. But if we have the gene of interest in between the beta galactosidase, this is the gene of interest, then the lacZ gene is disturbed or inactivated, so which will not form beta galactosidase, and we know the beta galactosidase is responsible for the blue color. So in these white colonies, the lacZ gene is disturbed by gene of interest, and then because one of gene of interest is there, that's why they are called as recombinant phages. So this is the first method, insertional inactivation of lacZ gene, which can be utilized for identification of recombinant phages. Now we have another method to identify the recombinant phages, and this method is insertional inactivation of lambda CL gene. Second method, insertional inactivation of lambda CL gene. Now this lambda CL gene is a special gene which is present at the map unit 38 in the genome of the lambda phage. Alright, so if this is the lambda phage and this is the DNA of the lambda phage, if we see the map unit, or if we see the map of this DNA, phage DNA, then at around 38 is the site, 38 is the map unit size, we will find a CL gene. We will find a CL gene in the DNA of this phage, lambda phage. Now, what is this CL gene all about? It is located at the map position 38 of the DNA. Okay, it is responsible for early regulation of the genes, and if we disturb this gene, then there occurs some morphological changes in the plaque. All right. So, what do? We cut this DNA at the CL gene. So, that means the CL gene is been disturbed now, and we insert the gene of interest in between the CL gene. So, we say that the CL gene here, the CL gene is inactivated. The CL gene is inactivated. All right. So, if this gene is inactivated and then the same process is repeated again, that means this DNA which is now modified is inserted in the phage, and the phage is allowed to infect the bacteria. The phages, recombinant phage, infects the bacteria in the similar way as we have seen before. The bacterial colony develop on the agar plate. This is the bacterial colony, as we have seen it before also. Then there will be bacterial colony on agar plate. All right. So, if we analyze the agar plate here, then there will be two types of colonies. Some of the colonies have got plaques which are very clear, but some of the colonies have got plaques of the phages where the phages have got some modified morphology, and then we call it them as turgid, or we can say that turbidity is there because of the change morphology. So, these are the clear plaques, but these plaques of the phages, they are turbid phages. The clear plaques and the turbid plaques of the phages in the bacterial colony are because of the CL gene. The clear plaques show recombinants. The recombinant phages. The recombinant phages are present in the clear plaques. If this gene is disturbed, then the phage morphology in the plaques of the bacterial culture are very clear. That means they have got a very clear cut morphology. But if the structure of the phages are disturbed, then it will show some turbid plaques, and this turbid plaques indicates non-recombinants. They are actually non-recombinants. So, what is the difference between the first and the second? In the first, we were analyzing the recombinant phages with the non-recombinant phages by color difference, but here by analyzing the morphological characteristics, we are identifying the non-recombinants from recombinants. The recombinants show clear plaques, and the turbid phages show they are non-recombinant phages. All right. So, these are the two methods which we have discussed here for identification of recombinant phages. Now, another method is there, that is we are numbering it as the third method. It is selection of recombinants or identification of recombinants using SPI phenotype. identification of recombinant phase by SPI phenotype. Alright, so what is this SPI phenotype actually? Suppose there is a bacteria and this bacteria is infected by a phage, and this is the DNA of the phage. So this bacteria is already infected by this is the bacteria. This bacteria is infected by a phage, and that phage is P2 phage, P2 lambda phage. Special strain is there. Now, this is the property of the system or biological system, that if a bacteria is already infected by a P2 lambda phage, then in normal phage, say for example, this is a lambda phage. Now, this lambda phage wants to infect this bacteria. This lambda phage wants to infect this bacteria. So, this bacteria will not take up the DNA of this lambda phage. Alright, so we say that this lambda phage cannot infect, it can't infect any bacteria with P2 DNA in it. That means if a bacteria have got a P2 lambda phage DNA, this is the DNA, that means this bacteria is already infected by another phage, then this lambda phage cannot infect this bacteria. Now, what happens if we design a phage in such a way, if we modify the DNA of this phage. Now, this phage is called as SPI positive. We call it as SPI positive. That means SPI means sensitive to P2 inhibition. Sensitive to PI is the strain of the phage. It is sensitive to P2 phage. That means if a bacteria already have a P2 lambda phage, then this lambda phage will be sensitive to this bacteria. It cannot infect this bacteria. That's why I call it as SPI positive. Now, what we have done here, we have modified its DNA by inserting our gene of interest. Say for example, if this is the gene of interest. So, we inserted the gene here. We inserted a gene here in this phage DNA, and now this DNA and this lambda phage become SPI negative. Okay. Now, if it is SPI negative, then this phage can readily infect the bacteria which have already P2 lambda. It can infect bacteria. So, that means we have inserted a gene. So, this phage is now recombinant phase. Now, this phage can infect the bacteria, and as we have seen in the previous also, then when the recombinant phase infects the bacteria divides and forms a colony. This phage also multiplies inside the bacteria and forms plaques. So, like before, we have got a agar plate, agar culture plate, okay. So, in agar culture plate, what do we notice here? That the plaques where the phages are multiplying, these plaques are present, these are recombinant phages. This plate is a recombinant phage. But non-recombinant phage, because the phages, the lambda phages which are SPI plus, they cannot even replicate. Alright, so they will die. They won't be able to replicate. So these plaques which are being developing is only of the recombinant phages, then so we can identify them. Alright, so like this we can identify the recombinant phages by SPI phenotype. SPI is the sensitive to P2 phage, and SPI positive are those lambda phages which cannot infect the bacteria. So we made these SPI positive into an SPI negative, and these SPI negative lambda phages are those phages where a gene is inserted into its genome, so its genome is modified. So now this phage can infect the bacteria and form its colony. So if the colonies are of the phages are developed in the bacterial culture, so these are the this is bacteria. Bacteria are also multiplying. These colonies will only be of the recombinant phages and not of the SPI positive or non-recombinant phages. Alright, and the last method which is adopted, that is to identify the recombinant phages, is the selection on the basis of lambda size, lambda genome size. Selection on the basis of lambda genome size. All right. Now here one thing is to be noticed, that if the proteins are organized in the form of phage, then the genome of the lambda phage should have approximately 37 kb to 52 kb size. So if this is less than 32 kb, then the DNA cannot be packed into the phages. Alright, so these are the proteins, and when these proteins are organized in the form of lambda phage, this lambda phage can take up the DNA from 37 kb to 52 kb. Especially if it is less than 37 kb size, then it cannot be packed. Alright, so what do? We take out the DNA and insert a gene into it to make it more than 37 kb or above, but up to 52 kb. So this DNA size is now increased. Now if this DNA size is increased, then we can easily pack this DNA into the phage, and this phage infects the bacteria, and these phages can be seen by the growing colonies. So these colonies develop. If any phage have got less than 37 kb size or more than 52 kb size, it will not multiply, the phages will die. But these colonies are of the recombinants, because we know we have made this DNA up to 37 kb size, and then these are the recombinant phage. These are the recombinant phage. And any phage less than 37 size cannot multiply. So in this medium, we will find the plaques with only recombinant phage. So this was the last method. So let me have a quick revision of what we have done right from the starting. So if we want to identify the recombinant phages, then we should know what are the recombinant phages. So if we take a lambda phage and we take out its DNA, we insert the gene of interest into it, and this gene of interest is put back into this recombinant phage, and then this recombinant phage infects the bacteria. This bacteria will multiply in a culture agar plate, and then because this DNA, the phage DNA is also present inside the bacteria will somehow lyse, and these many phages are released from inside the bacteria into the medium. So these round circles are the plaques. These are the plaques of the bacteriophages in the bacterial colony. So we say the plaques are present in the lawns of the bacteria. These plaques can be identified for whether they are recombinants or non-recombinant by using different methods. So for example, the first method is we know all the phages have got a DNA which have got lacZ gene. If we disturb this lacZ gene, then the beta galactosidase will not be formed, and the color will change into blue. So in the bacterial colony, the blue color are the normal phages or non-recombinant phages, and white one are the recombinant phages. Then second method we have seen, that was insertional inactivation of lambda CL gene. So lambda CL gene, that is CL gene, is a special gene present on the DNA of this lambda, and it is present on map around 38 map distance. Now if we cut this CL gene by restriction endonuclease and we add R gene of interest in it, and then this DNA is inserted into the phage. The phage infects the bacterial cell, and the bacterial cell forms colony. So this CL gene is responsible for morphology. That means if a normal phage develops, then it is having a clear, if the clear phages are there, they are recombinant phages. But if the phages morphology is disturbed and forms turbid morphology, then these turbid morphologies are the non-recombinant. So by analyzing the morphology, and the morphology is changed because of the disturbance in the CL gene. Alright. So identification of the recombinants using SPI phenotype. SPI phenotype is the phages which have got such a kind of phenotype. The bacteriophage lambda phages can be SPI positive, or it can be SPI negative. SPI positive are those normal phages which cannot enter into the bacteria, because if the bacteria is already infected by another lambda phage, P2 lambda phage, then it does not receive any other DNA. But if you want to enter this DNA, if you want to take this DNA inside the bacterial cell, then we have to modify this DNA. How did we modify it? We inserted a gene of interest in it, and then somehow this DNA has been modified. Now this bacteriophage will be called as SPI negative. This bacteriophage can infect into the bacterial cell. If the bacterial colony is developed, definitely they will be of the recombinants. And the last one is the size. If the size is less than 37 kb, then we have to we can add a larger DNA segment, that is a gene of interest, and then size is made more than 37 kb. 52 we do not touch, because we cannot, if the size is above 52 or near 52, then we cannot add our gene of interest. So we have to take the size to the lower limit. So that's why we are taking it 37 kb. So make it up to 37 kb by inserting our gene of interest, and then this genome is inserted into the recombinant phage. The phage becomes recombinant, infection with bacteria, and these colonies develop. Definitely these colonies will be the colonies of the recombinant phages. Other phages will die out. So thank you very much. Enjoy your day.